determining the population structure through maternal genetic lineages of the koala population of Port Stephens, New South Wales. A presentation by BrianTracey for transferable research methods. This research project started out as a growing response for greater research was discovered after completing my undergraduate research project. The purpose of this undergraduate research investigation was to supply a total estimated population count of the koalas of Tom Reed Peninsula of Port Stephens region in New South Wales, Australia. The project that I undertook stratified the total area into 70 half-acre plots in which day and nighttime surveys were conducted using various ecological survey methods. 2 daytime counts were conducted, first to grasp the basis of the area and relative population, and after this, subsequent nighttime surveys were conducted throughout the year in all 4 seasons to grasp the total estimated population. After the research was conducted and finalised, it was concluded that the population's total number had been severely underestimated, as the total number of individuals resulted in around 46. The population size resulting in 46 posed a few serious issues, as the initial estimation of the population was to be between 150 to 200 individuals. This isn't to say that all the koalas that were found in my investigation were the number that should be estimated, but it can be a little bit higher and it can be a little bit lower. So more needed to be done in this area. This population of koalas is a closed population, which means it has no area for migration or immigration of any other groups surrounding it. A population this small will closed off from other populations results in usually a high level of interbreeding. And interbreeding within a population this small poses a few serious issues, such as immunity depression, and which then can lead to disease outbreak within the population. This is where my master's research will come into play. Through the identification of all the genetic lineage of the population group within my undergraduate thesis, we will be able to determine the level of interbreeding through maternal lineage investigation. After discovering the maternal lineage, we will be able to see if there is a high level of interbreeding, and therefore be able to then investigate if the level of interbreeding is therefore reducing the overall immunity of the group, which is leading them susceptible to more disease. To study the genetic lineage of the population of koalas in Port Stephens, it will be done through sequencing the genetic samples that have been taken throughout the years by various organisations within the Port Stephens area. This includes research teams from Sydney University, local koala preservation societies, and local veterinarians who treat koalas that have been injured by roadside or pets around the area. The sample tissues that are being used for genetic sequencing have been taken from saliva swabs, fecal samples, blood vials, or ear tissue samples. All samples that have been taken throughout the years that will be used for the genetic analysis are being housed at a local vet service in Port Stephens, New South Wales. These samples will then be collected next year when it is time to research on them, and will be taken to UWF laboratory settings and where the research will be conducted. The sequencing technology that will be used was a next-gen sequencing machine such as the Iron Torrent. These samples will be each placed on a microchip, which will then be inserted to the machine and will be sequenced using 4 DNA nucleotides. After 3 hours of sampling, a receipt will be produced which displays the genetic makeup of each sample. Once all 100 samples have been tested, it will be then easy to match up the genetic lineage of all samples, as each individual will be compared once their results have been produced. The goal of this investigation is to determine the overall maternal lineage of the population of koalas at Port Stephens. This is to discover overall whether there is a high level of interbreeding, and then after that we hypothesize that the level of interbreeding is therefore reducing the overall immunity responses of this population species, as there has been high levels of outbreaks of disease such as chlamydia in the area. This is important to discover this because this population is in severe decline and all levels of conservation work that can be done should be done and will be discovered higher after this research project. The importance of this method in context of the proposed research project is quite significant. The method stated above uses leading methodologies for this investigation that are being done throughout the world in leading genetic studies. The machines and equipment being used is the most modern in the field, and they can, when combined with sampling methods out in the field used to take ear tag samples and blood or saliva, have all been tried and tested and have been proved through high literature in their field. Unfortunately, due to availability of the labs within UWS, as well as being able to access test samples in Port Stephens until later in this year, and a combination of not being trained on appropriate equipment has meant that I was unable to produce my own set of results this semester. However, this investigation has been taken further as I have been researching appropriate research literature throughout this semester to ensure that this research project is heading in the right direction. Tristan Lee, who is referenced in this literature, as well as his wife, are Australia's leading experts on koala genetics. I researched in depth these two people's papers as they produce at least 2 papers per year on the genetic analysis of Australia's koalas. For instance, this paper, produced in 2010, looked at a group of regional koalas in Sydney's area in New South Wales. These areas included the Blue Mountains and State Forest in the west. The results found that the group did follow the hypothesized results of my investigation, and by therefore having a low genetic diversity of a population that is small. However, this group that was investigated was a little bit different as it allowed for immigration and migration and was not closed like the population is in New South Wales and Port Stephens that I'll be investigating. Lee's next paper in 2012 is a paper produced in the field with similar hypothesized findings like my paper, which I hope to produce, in saying that small closed populations of koalas do in fact have a high level of interbreeding. This publication looked at koalas that do not live on mainland Australia and that are closed off as they live on islands such as North Stradbroke, Brampton and Rabbit Island. This paper investigated and found that the island population of koalas didn't have a strong genetic diversity like the hypothesized population in Port Stephens and was found to not be as healthy as mainland koalas. This paper strongly supports my hypothesized findings and will be used later as referencing for my investigation. Finally, Lee's paper that was also produced in 2010 provides an account of the genetic variation and structure of koalas in southeast Queensland. This source was investigated as it provides evidence of the population's habitat that is relatively similar to the Port Stephens koalas. Lee's paper targeted a group of a very larger scale containing over 510 individuals. Being much larger than the population of Port Stephens, the results can easily still be transferred. This group was found to have a high gene pool which could put down to the large group or just simply how well they adapt to their surroundings. This is an interesting paper to look at as it did have some similar differences, but then can also be related to the hypothesized theories that I will be investigating.